Hello, my name's Neil Spooner, Chairman of the Patient-Centric Sampling Interest Group. I'd very much like to welcome you to our February webinar, which is Perceptions and Acceptability of Microsampling for Remote Monitoring in Patients with Plasma Cell Dyscrasias. I can't say that. Brought to you by Nithya Paranthaman, the Senior Scientific Officer at the Royal Marsden Hospital. Sorry, I'm really tripping over my tongue today. Before we get started, just want to take a moment to thank all of our partners and sponsors who provide funding that enables us to have these webinars and carry out the other activities of the Patient-Centric Sampling Interest Group. If you have questions for Nithya, please submit them via the chat function. There should be plenty of time at the end of the webinar to go through those questions. I'll read them out. And just a reminder to everyone that this webinar is being recorded and in the next few days will be available through the PCSIG YouTube channel and also through the evidence base and the PCSIG websites. So without any further ado, I'd like to hand over to Nithya. Thank you.
Thank you so much, Neil, for the introduction and inviting me to be a part of this webinar series. I'm just going to go ahead and share my screen. So hopefully you all can see my slides now.
Yeah, that works, Nithya.
Okay, perfect. So again, my name is Nithya Paranthaman. I am based in the Clinical Biochemistry Department at the Royal Marsden Hospital. And so I thought today, rather than giving you all a technical talk, I'd come on here and talk about the multiple myeloma patient story. And discuss how we incorporated patient and public involvement into our clinical trial. So just a little bit about myeloma before we get started here. Myeloma is a blood cancer that originates in the bone marrow. Cancerous plasma cells differentiate uncontrollably, and that leads to an overproduction of what we call monoclonal protein, also known as paraprotein. So there's an estimated 6,000 new diagnoses annually in the UK. The median age of onset is 72 years old, and it's more common among males than females. So although treatment for myeloma has significantly improved over the last 15 years, improving prognosis, unfortunately, myeloma is still considered to be incurable. So it is a relapsing remitting cancer. Patients will experience periods of active disease followed by remission. So it's so important that these patients are continually monitored. And one way they are monitored is through simple blood tests. So laboratory assessment of myeloma includes serum protein electrophoresis. This is done to detect M protein. If M protein is detected, a fix can then be done to characterize the subtype of myeloma and determine clonality. Additionally, intact immunoglobulin and serum-free light chain levels are also measured to give clinicians a better indication of the disease state. It also lets clinicians know whether or not a patient is responding well to treatment, and also if they may be potentially relapsing after a period of remission. So again, these blood tests are crucial for the monitoring of multiple myeloma patients. Living with myeloma presents with certain physical and mental challenges. Some of the difficult symptoms that patients face with is fatigue, peripheral neuropathy, steroid-induced effects, which can include mood swings, difficulty sleeping, and even hallucinations sometimes. Bone pain is also a very common symptom that these patients face. So, what we hear constantly from patients is that they learn to live with these symptoms. Although myeloma is treatable, they do have to deal with these difficult symptoms on a daily basis. And for some, it can be quite debilitating and make everyday routine tasks even more harder. So the current patient pathways within the Royal Marsden Hospital, patients will either come in a day or a few days before to have their bloods drawn. And then if they're not having a phone consultation, they're required back into the hospital to have their face-to-face consultation or other appointments such as treatment. The second pathway, bloods are done on the same day as other appointments that patients may have. So the difficulties with these pathways is that the most recent blood results aren't always readily available for the patients to discuss with their clinical care team. Patients are having to wait until their next appointment, which can be weeks or months away, to discuss these results. And then also, if they are, having other appointments that week for treatment or having an in-person consultation with their clinical care team, they're having to make additional trips to the clinics just to have their bloods drawn. So with this specific blood test, especially the intact immunoglobulins and the serum-free light chain assays, once a patient is baselined using this particular assay, it's recommended that they are continually monitored using this assay. So it's not like they can go to their local GP or local clinics to have their bloods drawn. They do unfortunately have to come to the Royal Marsden for their bloods. So I have a quote at the bottom of this slide from one of our PPI contributors. They asked for their bloods to be taken first thing in the morning, but as with all phlebotomy clinics, waiting times vary. So sometimes you're in there for a few minutes, or sometimes you're waiting hours just to have your bloods drawn. And then this patient also highlights the difficulty of not having their most recent blood results readily available to discuss. That just adds, another layer of anxiety and stress for these patients that are already dealing with a difficult disease. So we wanted to understand the patient story a bit more. So we surveyed a group of our patients that regularly attend the RMH clinics. 71 patients returned a completed questionnaire. So over half of these patients attended clinic that day for phlebotomy services only. Again, if they don't have a phone consultation and are required back in, that means that they're making these additional trips within a very short time frame, usually within a week. More than a third of these patients had someone drive them to their appointment. So not only are they having to rearrange their schedule, but a loved one or a spouse is having to rearrange their work or social commitments to accommodate these very frequent appointments. And a one-way journey to the Royal Marsden Hospital took patients on average 61 minutes. So we are a tertiary cancer hospital, so we see patients from all over, ranging from 3 to 146 miles away. So if they are, on active treatment, making these journeys to the hospital very frequently can be tiring. We did ask these patients a few more questions. We wanted to see how they found traveling to the hospital for routine blood tests, and if traveling to the clinics for blood tests hinder their social and work activities. So how they found traveling to the hospital, the overall rate was a 1.76 out of a 5, so mild difficulties there. Does traveling hinder their social activities? Overall rate of 1.86, so mild inconvenience. And for those that are employed, does traveling to clinics hinder their work activities, and an overall rate of 2.75, so moderate inconvenience there. We were a bit surprised with these results, but I do want to say that the survey was taken during one of the lockdowns during the pandemic. So again, a lot of these patients are elderly, and they were at that time isolating themselves in quarantine. So coming to the hospital was actually a bit of an outing for them, and a chance for human interaction. So we're interested to see if these answers change now that we're out of the pandemic, and we will be surveying our patients again. So the overall aim of our research study is to incorporate microsampling into the routine clinical care of myeloma. We're hoping by doing so, this will reduce the number of trips that patients have to make to the clinic. This gives patients an at-home option of collecting their blood samples autonomously and shipping that back to the hospitals. And then also with this scheme, if patients do send their samples to us, our labs, well in advance, then their most recent blood results may be readily available to discuss with the clinical care team. And because this is such a patient-focused clinical trial that we are conducting, we thought it would be most appropriate to hear patient voices and hear their opinions and perspectives on our study. So we decided to integrate PPI, patient and public involvement, very early on into our clinical study. So the NIHR defines PPI as research done with or by patients and the public, not to, about, or for them. It's working collaboratively with patients and the public and sharing decision-making. So there are different ways in which you can incorporate PPI. You can incorporate it at various stages, but we decided that we wanted to have a PPI panel. So we established a national PPI panel with 14 members, 6 of them male, 8 female, ranging from 35 to 78 years old. Most of the panel members do have lived experience of multiple myeloma, with one being a caregiver. So the way that we went about recruitment, we contacted Myeloma UK support group leaders. So these support groups are situated in various areas across the UK. We discussed our trial with them. We told them about these potential PPI activities and what we hope to achieve. And they distributed that information to their support group members, and interested individuals reached out to us if they wanted to be a part of our research study. We did try to apply snowballing tactics to reach those who are seldom heard, less reached, or ignored individuals. Out of the 14, we have 4 members from these communities. Obviously, this needs improvement. We do want a wide range of patient voices in our research, and this is something that we are trying to improve, and it's a work in progress. So when it comes to microsampling, we are fortunate in this day and age where we have a variety of microsampling devices out on the market. But for us, as a NHS clinical setting, we did have a few criteria, such as low cost, easy to use, and a device that would be easily embedded into our laboratory workflow. So we narrowed the list down to these devices on the screen here. So we looked at, we're looking at just traditional filter paper. We're looking at a group of devices, a quantitative dried blood sampling devices such as the Hemaxis, the Capitainer B50, and the Neoterix meter device. We're also interested in some of the newer upper arm devices, such as the TAP Micro and the Tasso. And although these are a bit more expensive than the other devices out on the market, it would be liquid blood, capillary liquid blood that we would be collecting from the upper arm. So since it's not a different matrix to the matrix that we're currently using, we wouldn't really need an intense method development and validation if we were to use these devices. So that would save us a lot of time and resources. So that's why we were interested in those devices, even though they're a bit more expensive than the others. So we are doing preliminary work with these devices in our lab and in our clinics, but we also wanted to hear what our panel members thought of this and their initial reactions to these devices. So we conducted a series of workshops. Our first focus group workshop was very much inspired by a group up in Liverpool who sent out mock sampling kits of the VAMS Mitra devices to their cohort of interest. So we did something very similar. We sent out mock sampling kits of the devices that we were interested in, and we also sent along aliquots of diluted maple syrup with red food coloring to mimic blood, and also a pipette. So for the first half of the workshop, I went through what our research study was about, and then gave our panel members a bit of background of these devices that we were interested in and also showed them how they are used. And so for this initial workshop, patients weren't using these devices on themselves, but rather using the diluted maple syrup that we had provided. So I talked them through using these devices and collecting samples using aliquots of maple syrup. And for the second half of the workshop, we had a discussion on just their initial reactions to these devices. So it was quite successful. Our second focus group workshop, we had our panel members review and edit all of our patient-facing documents. So this includes the questionnaires, the patient information sheets, and the consent forms that we will be using, or that we are using in our clinical trial. We also wanted feedback on our trial protocol. So we shared our trial protocol with our panel members and specifically wanted input on how best to go about recruiting patients into our trial and how best to go about collecting samples. So again, a lot of useful information from this workshop as well. We conducted both of these workshops online through Microsoft Teams with the permission of our panel members. These meetings were recorded. The panel was encouraged to enable their video cameras to optimize engagement and the interaction. And what we found was that our panel was very enthusiastic and very, willing to be a part of these discussions, which was super helpful and valuable to our research study. We did award our panel members. So for the workshop, they received £20 for their time and efforts. And for the second workshop, we did award them with £40 just because it was more involved. We had sent them our documentations beforehand to give them time to review and edit. So just outcomes from our first focus group workshop. The overall consensus was that microsampling would be a welcome addition to the routine clinical care of myeloma. Initial reactions were generally very positive. The important factors that the panel brought up during the discussion was ease of use, reliability, sustainability, whichever device that we move forward with. They hope that it was accurate, precise, low cost, and that it fits through the letterbox. So I just want to go through a few of the comments and a few of the discussion points that came up during our first focus group workshop regarding these different devices. So regarding the traditional filter paper, I had patients use the pipette to drop drops of the diluted maple syrup onto the filter paper. And with that exercise, patients could see that, these spots were of varying sizes, varying volumes, and not all they didn't all look the same. So we did discuss that, if we were to bring this into the clinics and we use traditional filter paper, this is one of the difficulties. We do see traditional filter paper in clinics where patients have returned these devices with varying spot volumes, and it's a bit more difficult for the lab to use these samples. So we went on to discuss the hemaxis. So for those that don't know, this is the hemaxis device. Hopefully you can see that okay. There are 4 channels at the bottom, and when used correctly, these 4 channels will wick up 10 microliters of blood, and you will see 4 equal spots at the top here. So patients or our panel members really appreciated the fact that they would be able to visually see these dried blood spots when used correctly. However, one of the difficulties that they mentioned when using this device was that the channels at the bottom were a bit difficult to see. And again, we are working with an elderly population here. With age comes difficulties with eyesight. So that was one of the concerns that was raised using the hemaxis device. So moving on, talking about the Capitainer B50, the panel really appreciated the fact that this was a sustainable device, paper-based, where the other devices used some degree of plastic. The channels did protrude out, so it was easier to see. But one of the difficulties that was mentioned with this device was that they didn't know when to pull away. So they weren't really sure of when there was enough sample that had been collected using this device. The Neoterix meter device was actually quite popular with our panel. These are handles with absorbent tips. It was easy for the patient for the panel member to collect the sample. However, again, they did mention issues. We did discuss issues of undersampling and oversampling. So with all of these devices, proper training and practice can overcome the difficulties that were mentioned. Patients also did have a look at the upper arm devices. This is the TASO and this is the TAP here. Their initial reaction when they did see these devices was that, oh, this looks scary. This is not something that, I'm going to first reach for when given a variety of devices. But one of our panel members is a diabetic, and she uses a continuous glucose monitoring device, so a CGM device. And she mentioned that, after that initial psychological hurdle, these devices are essentially painless, virtually painless, and we discussed how the technologies are the same. So by the end of this discussion, the panel was really warming up to the idea of these devices, which was great, and which was brought about with that important discussion session. So again, with any of these devices, training is going to be key and something that we will be focusing on as this trial progresses. So for the second focus group workshop, again, our panel reviewed and edited our patient-facing documents. There were a lot of good suggestions for how to improve these documents. They suggested that we reduce the number of medical terms because it could be off-putting and slightly frightening. So we've taken a lot of that out. They did suggest to make the documents shorter, as they didn't want to, if they were to sign up for this trial, they didn't want to sit there and read through pages and pages of wording, and they'd want it to be a bit more clearer and more concise, which we've taken on board as well. So with regards to the actual trial protocol itself, the focus group members said they would sign up for this trial if they were asked to do so. They didn't think that we would have any difficulties with recruitment. They said incentives wouldn't necessarily be required, although if reward payments were provided, it might be something that could potentially retain our recruited trial patients for the entirety of the study. So again, lots of valuable information here. A lot of things that we may not have been able to pick up on if we didn't conduct our PPI activities. So some comments from the discussion that I wanted to highlight, the first one being anxious to help research. When we first set out to do these PPI activities, we weren't really sure how the turnout would be and what sort of response we would get, but we've gotten such a positive response, and we've learned that, a lot of these myeloma patients, individuals with lived experience of myeloma, are willing to be a part of research or willing to, have their voices heard, and really would like to be a part of clinical trials, which has been a very pleasant surprise. When collecting samples myself, I might find it less easy than when someone else does it. Practice would probably overcome this. Only small concern would be self-administering finger prick tests. I would do it, but would be slightly less preferable to having it done, but I get it. So for this first phase of the study, I will be down in the clinics collecting patient samples, capillary patient samples, so the finger prick samples as well as the upper arm samples alongside their venous bloods. And so, again, this just goes back to that initial, psychological hurdle of pricking your finger or pushing that button on the upper arm devices. And like this patient says, practice would probably overcome this, and it typically will. The next quote, I need reassurance that the quality of results from the finger prick is as good as from the traditional test. So we are a clinical lab. We do have certain standards that we adhere to. So we did provide that reassurance to our panel members that if we were to bring a microsampling scheme into clinics, we would have done, all those studies on precision and accuracy. We would never bring a method into the clinics if it didn't pass all of those tests and meet a certain standard. Friends live nearby the hospital, so we go and see them afterwards. So doing a finger prick test at home would lead to that loss of social contact. So again, we're not totally eliminating all of these visits to the hospital. We're just trying to reduce the number of visits that patients have to make during this patient pathway. Drawing blood in this way is always difficult for me as my hands are cold and blood flow reduced. So we haven't had too many difficulties in the clinic so far, but we do try to tell patients to, warm up their hands, do some circles with their hands, with their arms to increase that blood flow and circulation. Also, with a lot of these microsampling devices, companies within their kits, they're providing these hand warmers, these warmers. And so, it could potentially use this if we did see an increased issue with our multiple myeloma patients and difficulties with collecting their capillary blood samples. So I have a video from a panel member here, and she just describes her experience of being a part of our panel. And so I will go ahead and play that now.
I'm Cecilia. I've been living with myeloma since September 2020. I became involved as a patient to provide our patient perspective with regards to some work that was being done to look at blood sampling at home. So where we as patients might be able to take the bloods at home and post them in rather than having to go to the hospital. And this is, great. I really enjoyed being involved in this because we sent all the different types of devices, and then we were shown how they worked, and then we used them as well with syrup with food coloring, so fake blood, so to speak. And it was quite interesting to try all the different devices, and then everybody gave their opinions on which ones they felt was the easiest to use, which one they felt would be easiest for patients. We also took some time to review some of the documentation that would go out to patients who would be signing up to participate and take the bloods at home. And yeah, it was great. I enjoyed being involved. I enjoyed the interactive aspect of it, and also meeting other myeloma patients, but also the fact that feeling that our voice is being heard from a patient perspective. I think that's vital, that sort of perspective is brought in to any piece of research. So yes, I do feel our voice is making a difference. Everything we said was well taken on board, and I'm sure that it will help enhance the experience for other patients who do eventually take part in the sort of physical part of it when they get going with the testing and being able to take bloods from home. So yeah, that's it from me. Thank you.
So again, 2 very successful workshops. We are planning for future workshops. Our next PPI workshop, we're hoping for this to be a face-to-face workshop, trying to find an accessible event space to conduct it. For this next workshop, we will be focusing on the training aspect of these devices. So our PPI contributors, our panel members, will help in reviewing and designing our training material, because that is very crucial for the success of a microsampling scheme. We are hoping to reach out to different communities, make sure their voice are heard as well, and invite them to this PPI workshop. just planning ahead for culturally appropriate food and interpreting services if needed. All patient incurred expenses, travel and care costs will be covered. And as this will most likely be a half-day event, our panel members will be awarded £75 for their time and efforts. So a few more workshops lined up in the pipeline that we are really quite excited about, because the response so far has been very positive and we've gained a lot of knowledge from it. Some practical considerations, some of the things that we thought about before conducting these activities. Payment and reward, like I said, we are rewarding patients or panel members for their time and efforts. Logistics, before we schedule these activities, we do ask them, what days, times, and lengths of the meeting would work best for them. And a lot of our panel members said that, they had clinics earlier on in the week. So we've been conducting these workshops on a Thursday from 10 to 12, as they did say that with myeloma, their energy levels decrease throughout the day and they have the most energy in the morning. So we've had these workshops in the morning for 2 hours, 10 to 12, with a comfort break, which seems to work really well with our panel members. Consent, the beauty of PPI is that you don't necessarily need formal ethics, but you do need to conduct your PPI activities in a very considerate and ethical manner. We had agreement forms and project summaries and different documentation that we provided our panel members that explained what they would be doing, what we'd be doing, and what we'd be doing during these workshops. And that leads me to my next point of safeguarding. All of this work is voluntary. Patients can decide if they want to be a part of a workshop, and if they don't, they are free to withdraw and join whenever they'd like. So safeguarding panel members' health and well-being, and making sure that their personal details and data, all that is protected. And also recognition and appreciation. We have learned so much by doing these activities, and we're truly grateful for, our panel members' time and, being involved with our activities. So we do try to recognize that and send them, our heartfelt appreciations. And I hate that I keep referring to them as patients and panel members, because over the course of last year, they've become our colleagues and collaborators on this project. I think when Neil reached out and asked me to be a part of this webinar, I think it was about a year ago, when we had just started looking into the PPI activities and looking to embed PPI into our trial. And so I said back then that I'd talk about PPI, hoping that we'd achieve something by then. But a year on, we've had successful workshops. We've recruited a national PPI panel. So it's been quite valuable. So hopefully Neil can invite me to speak or give a webinar again in a few years' time, and hopefully I'll be able to tell you all that we've successfully embedded microsampling into the routine clinical care of myeloma. But I really just wanted to encourage you all to think about myeloma and see where you can implement, think about PPI and see where you can implement PPI into your research. And if not, just maybe, become more aware of PPI and do some research, and maybe even attend a training session. So for those that are based in a a hospital or a trust, typically they will have a PPI department or a team that you can reach out to. So I encourage all of you all to do that. And I'm also willing to help. If you'd like to contact me for more details, or if you'd like to see our agreement forms or the documentations that we've come up with, for our PPI panel, I'd be happy to share that as well to give you all some sort of a template. So yes, please do reach out. So just a few slides here to wrap up challenges and lessons learned. Recruiting a representatively diverse focus group, this is definitely something that we are working on. One of the suggestions from our panel members was to reach out to community leaders. So right now our panel is very heavily patient based. So a lot of individuals with lived experience of multiple myeloma, but maybe reaching out to various community leaders would diversify the voices that are brought into this research study. Digital exclusion, as this is a national panel that we created, our first 2 workshops had to be done online. This was done via Teams again, and so only those that had experience with using a computer and using Microsoft Teams were able to join. So we would definitely like to make this more inclusive and accessible to a wider range of individuals. So hoping that our next workshop, which will be face-to-face, will provide that. And then effective PPI requires expertise, training, and adequate financial resourcing. PPI is just a puzzle to the entire jigsaw, or small piece to the entire jigsaw puzzle that is microsampling. So obviously there's the whole technical side of things, and we're liaising with clinicians and the regulatory side of things. But I think PPI at this point is not necessarily a nice thing to do, but something that's required and needed. And, we have a very good PPI team at the Royal Marsden Hospital that we'd reach out to when we needed any advice. We also attended all of their training sessions. And we were also awarded a grant by our PPI department within our hospital to do these PPI activities. Without that grant, I'm not sure if we would have been able to do these in-depth PPI activities. So very grateful for that funding. Then lessons learned. Myeloma patients are very eager to join research. They're anxious to join research, as one of our panel members had said. So this was definitely a pleasant surprise, and it has really shaped the way our research study is going and the design of our clinical trial. Focus group participants can clearly see the impact of patient-centric sampling can have on the multiple myeloma patient pathway. So again, a lot of positives that have come out of our PPI activities. And I think as researchers, oftentimes we are stuck in the lab for hours at a time, sometimes even with minimal human interaction. And we're just so consumed with these hundreds of samples that we forget that on the other side of this, there is someone desperately waiting for those results, for that data. So incorporating PPI and being able to work with a panel with lived experience of myeloma has really been eye-opening for me, and it has really shaped the way our project has been going. So I'd like to end with a key message here. Involving myeloma patients at an early stage has ensured that this trial has been shaped by the lived experience of multiple myeloma patients. So this is the end of my presentation, and I am happy to answer any questions. Thank you all.
Thank you, Nithya. That was a fantastic presentation, very uplifting, very informative.
Thank you.
And yes, we'd love to have you back in a year or so. And next spot is something like May 2026. So we can talk about that later.
Yeah.
But yes, we'd love to hear more about what's going on. So we've started to get a few questions coming in. Just a reminder to everyone, if you can submit through the chat function, that would be fantastic. So the first one came in from Paranthaman, who I think you might know Paranthaman quite well.
Yes, that is my dad.
Work. Is it possible to extend this clinical trial to other forms of cancer?
Definitely. I think first it's best to start with a specific cohort of patients. And as we learn from this trial, we will definitely be looking to expand microsampling into other cancer schemes. So yes, definitely in the works.
Thank you, Nithya. Question from Frederick. We're just planning a study. With your experience, would you recommend us to use upper arm device and capillary tube or finger prick, lancet and DBS? The patient group contains women aged 40 to 65.
Yeah, so I mean, there are just so many things to consider here. You know, which analytes you're looking at, and also budgeting for this study. Like I had mentioned before, the upper arm devices collect liquid blood, so you wouldn't necessarily have to perform a detailed clinical validation and development, which saves a lot of time. But if you are looking for a more cost-effective device, then would suggest looking at one of the quantitative dried blood spot devices. So it's very patient specific as well as project specific and analyte specific. But yeah.
Thank you, Nithya.
Further.
Question from Bill. Have you determined the limitations of the laboratory assays with regards to the various collection devices and sample types?
So yeah, this is what we're working on, the preliminary studies, the amount of volume that we can get from these devices, and if we're pushing our instruments to the limit. So yes, this is something that we're currently working on and trying to determine.
Yeah, and hopefully there's going to be more guidance out there to help you with that in the future as well.
Yes.
Question from Elizabeth. Really interesting work and a great example of PPI integration. Were you surprised that so many people would be keen to do self-sampling? It seemed that most participants were. Do you think there's a risk that you got a bit of a positive bias, i.e. people who volunteered for your PPI group would be, by virtue of being interested in research, more likely to embrace this sort of technology?
Yeah, no, thank you, Elizabeth. Yeah, we were actually quite surprised by the response that we received. A lot of these patients, panel members have had past experience with finger prick sampling. So this was something that they could see easily embedded into their patient pathway. As far as the positive bias, again, we are really trying to get a diverse set of patient voices, looking to get members of those communities that are less heard, less less reach. So once we are able to get, a more diverse set of voices and stories, hopefully, we'll get a better understanding of whether or not these patients would be willing to do something like this in their homes.
Thank you, Nithya. A question from Richard. Are you planning to share this important work with other trusts?
Yes, we'd definitely like to publish our results. And again, if you'd like to reach out, I'm more than happy to go into further detail and share with you all what we've done and what we plan to do. So, yes.
Thank you, Nithya. I have another question, and you sort of skirted around it a little bit during your talk. Mm-hmm. So you don't require ethical approval to run these kinds of these PPI panels.
Yeah, so the survey that we did initially, that requires ethics, but these PPI activities that we did with our panel members doesn't necessarily require ethics, as we're not recording any information or medical history of the panel members themselves. They're essentially just advising us, collaborating with us, rather than being a part of the study, if that makes sense. So don't need ethics. There is a tool on the HRA website that allows you to see if your particular PPI activity will require ethics or not. So if you're unsure, I recommend you look at that tool.
Okay, maybe we can include a link to that tool on the PCSIG website in the future, along with maybe some of the other materials you're talking about here. I think it might be quite interesting, Nithya, to generate a page that helps people with this PPI piece, with some guideposts.
Yes,
we can talk
definitely.
about that later.
Yeah, I'd love to be a part of that.
So are there any broader guidelines? You talked about the HRA website, which I assume is UK based.
UK based.
Do we know of anything else internationally that is available to people, or do people tend to go to this HRA website?
So, I mean, as I'm currently based in the UK, and this is UK-based study, we looked at the NIHR website as well for guidelines. The Oxford PPI group also has a great website with some really good advice here. In terms of internationally, I haven't quite looked at that, but I would assume it's pretty similar. But something that I could reach out to colleagues in the States or do a bit more research on if we were to build this page. So,
yeah, and I think, well, patient-centric sampling interest group has its own patient advisory group, and also planning to do a lot of work with PIC United, who I think are present on this call as well, and maybe we can work together to build some content for the website that would be useful internationally to help people considering this, because I think it's absolutely pivotal to get this patient involvement and get that patient voice involved in our studies. In fact, some of the regulatory bodies are now for clinical studies. So I think people are wanting to learn a lot about this. I'm not seeing any more questions. So I'm just going to, if you don't mind, Nithya, I'm just going to share a couple of slides.
Yes.
I'd just like to say thank you before the slides. That was, we've got a last minute one here. No, I don't understand the additional question, Frederick. I don't know whether you want to put that to us again or after the event. Anyway, so I do have some slides I'd just like to share at the end here. Let's just put this content. So just a reminder that we've got another webinar coming up next month on March the 11th from Alexandra Kitts, talking about saliva as a diagnostic tool for advancing accessible testing. So I'm really excited about this next webinar, because patient-centric sampling isn't just about blood, and there are other biological samples that are available. So please take a look at that, and please register for that webinar that's coming up on the 11th of March. For those that aren't aware, the patient-centric sampling interest group, I'm not going to say too much about it, but if you use those QR codes, the one on the left will take you to our website that has a lot of information. The one on the right will take you to our LinkedIn group, which is a community of over 500 of us now, who are all passionate about breaking down the barriers for patient-centric sampling, educating more people, and understanding what the barriers are and figuring out how to overcome those. So we've got a bunch of working groups. Take a look at that, and anyone that wants to help us, you're more than welcome. So with that, I'd just like to thank our partners and sponsors again. As I said before, without them, we couldn't do what we do, the Patient-Centric Sampling Interest Group. But most importantly, I want to thank you, Nithya, for a really fantastic presentation, really good Q&A session, and really looking forward to inviting you back in a year.
Thank you, Neil.
I have to say. And thank you to all our audience today. We had a great session today, and speak to you all soon. This video will be available on our YouTube channel and on our website in the next few days. Thanks all.
Thank you.